Hello, everybody. How's life? How's things? Hope everyone's doing well. Hope everyone's staying safe. We are going to kind of continue with the urinary system, right? If you will recall, right, last presentation, right, we had gotten into really just talking about the nephrons, right? That unit, that structure which produces the urine. Right? If you recall, there was the renal corpuscle and there was the renal tubule. The renal corpuscle, that's what was responsible for the actual production of the filtrate. Remember that filtrate? That's what will eventually become urine. But before it becomes urine, we can modify it. That modification, that all takes place along the renal tubule, and that's where we had gotten to by the end of last class. So, again, the renal tubule, that's this part here, this squiggly, right, these squiggly lines of stuff. Right, that is the renal tubule. Now, there's three things, or kind of three parts of it. Right, there is the proximal convoluted tubule. There is the loop of Henle, or the loop of the nephron, and there is the distal convoluted tubule. We're going to break each one of those apart here in just a minute, because depending on what you are, whether you are glucose, amino acid, sodium, potassium, water, right? Certain parts of the tubule allow for you to cross the wall, right? So certain parts of the tubule are impermeable to water, for instance, right? Certain parts of the membrane are impermeable to sodium. That's where the hormones come into play. The hormones are the key. They are the influencer. They are what can actually influence how permeable or how easily some of these molecules can get through the tubule. Now, kind of two terms that we want to kind of think about or kind of understand: reabsorption and secretion. Alright. So do we see how surrounding the tubule, right? This is all tubule through here. All tubule. All this tubule, this kind of brownish tube. We see all this capillary bed, right? This kind of webbing meshwork of capillaries. Those are the peritubular capillaries, or what are called the vasa recta. So when we talk about, I mentioned, our body can modify what is in the filtrate, right? Our body can modify, or our renal tubules can modify what's in the filtrate. So let's say if I'm in the tubule, certain parts along the tubule, right, our body can reabsorb, let's call it glucose. So if we reabsorb it, that means it leaves the tubule and enters into the bloodstream. Cool. If we talk about secretion and say, our body can further secrete sodium or potassium into the tubules, what that means is, let's just stick with sodium for instance for a minute. That means sodium would go from the capillary and enter into the tubule, right? So whereas reabsorption, our body's reabsorbing from the tubule, meaning from the tubule back into the bloodstream. And if it's secreting molecules, that's when it goes from the vasa recta, from the capillaries into the tubule. Alright, so just kind of keep that in mind as we go through these next few steps. So the first part of the tubule, right, something called the proximal convoluted tubule. It should say 80%. For some reason, I don't know. Apparently, I had something against the percentage sign. I don't really know, but it should be 80%, right, of the filtrate is reabsorbed here. Meaning, of all the filtrate that ends up in Bowman's space, Obviously, that would be 100%, right? Because that's where all the filtrate ends up. 80% of it, 80% is reabsorbed here in the proximal convoluted tubule. That means in all the rest of this, there's only 20. Right? And now I say, actually, then let me back up. It's actually not even 20. Obviously, that would mean we would produce no urine. So that actually, that number is incorrect. Right? Here, throughout the entirety of the rest of it, loop of Henle, distal convoluted tubule, the collecting duct, right? Depending on our hydration levels, right, there might be as much as little as 5%. There might be as much as 15%, which is reabsorbed. Right? I was just trying to do some simple math, 80 and 20, but I forgot there for a minute that not all of the filtrate is reabsorbed, because if it were, we wouldn't produce any urine. Right? So, anyways, the proximal convoluted tubule, the PCT, that is where, again, the vast majority of reabsorption from the tubule occurs. And you can see glucose, amino acids, right, a multitude of ions, water. Yeah, there's really very little that the proximal convoluted tubule is impermeable to. It is for all intents and purposes, it is permeable to everything. Right? Amino acids, glucose, right, sodium, potassium, etc. Right? We can see one of the things which maximizes this reabsorption, right, is something called a brush border. Now, we talked about something like this when it came to the intestines. Right? If you remember, lining the inside of the intestines, right? There was the villi, then there was the microvilli, right? And that's what developed, or that's what was a part of the brush border. The brush border is where we maximize the absorption. Remember, the only way that our body can absorb or reabsorb nutrients, right, is if that nutrient comes into contact with the wall, right? Like with the intestines. The only way we could potentially absorb the vitamins and minerals that we consumed is if they then came into contact with the actual wall of the intestine. So we had to maximize the surface area of the intestine. So that's where the brush border, again, the brush border is microscopic, right? I'm talking, right, there's thousands of villi, and then they're lining the thousands of villi are thousands upon thousands of microvilli. Right? So we're talking about microscopic scale, right? Many more magnification than what you have ever seen in the school setting. Right? That's how small we're talking about with the brush border. Right? We saw images of that along within the digestive system. Remember, one of the things that we are going to kind of take a look at, right, is this right here. Right now, this is just kind of a general view of what takes place, of what goes on along the proximal convoluted tubule. Because so much reabsorption takes place, right? You can see, right, one of the more important aspects of the proximal convoluted tubule is that some of the reabsorption occurs via active transport. If it's active transport, remember, that means it's going to utilize energy to actually reabsorb those particular molecules. Right? So, within that, there's a couple of things to look at. Right here. Right on this side. Something called a symporter, right? There's ways that we can reabsorb and secrete molecules. Right? There's a way we can reabsorb and secrete sodium, potassium, glucose, amino acids, etc. It's not like we can just say, oh, hey, that glucose is supposed to be in the body. All right, let's reabsorb that glucose. Oh, hey, that's sodium. We want that back. Let's reabsorb that. It doesn't work exactly like that. Sometimes it's Peter for recall, or sometimes you can reabsorb multiple molecules at once. When we absorb multiple molecules at once, that's called a symporter. Right? Molecules traveling the same direction. Right? When we look, right here is in here the lumen. This is within the tubule. So anything that remains in this tubule here is going to become urine. Right? So here's the tubule. You can see sodium and glucose are reabsorbed together. If they in order for sodium or in order for glucose to be reabsorbed along the PCT, right, this symporter takes place. Now you can see move down here, movement of hydrogen as well as bicarb greatly influence pH. Well, yeah, right. We're going to talk about that, right? Over here, we talk about the movements of hydrogen. We talk about the movements of bicarb. That's what can influence the actual pH of the blood. Remember, pH 1 to 14, right? Neutral. 7. Right? Anything that way is acidic. Anything that way is basic. All right. Okay. So, this here, right? This is certain medications, certain diabetes medications. Well, God, what is it called? Not Clopidogrel, Invokana. Right? One of the medications, it deals with blocking or preventing the movement of sodium across the PCT. Right, so let's think about this for a second. If I am an uncontrolled diabetic, that means I have got more glucose in the filtrate than it's supposed to be. Right. If I'm a diabetic and I've got more glucose, more blood sugar than I need, one of the ways these medications can work, and specifically Invokana, well, I don't want that glucose to re-enter the blood if I've already got high blood sugar. I want it to remain in the urine. Right? I want it to remain in the tubule. Because remember, if it remains in the tubule, that means it's going to eventually end up in the urine. Right? So by this diabetes medication, Invokana, that blocks the reabsorption of sodium. Well, that's a symporter. Oops, that's a symporter. So if we block the reabsorption of sodium, then glucose can no longer cross the tubule wall. Does that make sense? All right. Let me kind of explain, normal, physiology first. Probably should have started with that. But just in normal physiology, right? We want as much glucose as humanly possible, right? That's why when it comes to the intestines, that's why when it comes to these tubules, right, we want to absorb as much glucose as possible, right? Glucose, that is the molecule of life, everything from the bacteria to the leaf outside to the cockroach to the to whatever it is to us, right? Which can sometimes be confused with the cockroach, but anyways, right? We want to absorb as much glucose as possible. Until we don't, like in diabetes, but we'll get to that in a second. So, recall how I mentioned, right, in the glomerulus, right, the composition of the filtrate, right, this fluid which is filtered out of the glomerulus and captured in Bowman space, has a very similar concentration to that of the blood. Similar, right? It has many of the same molecules. Glucose, amino acids, potassium, calcium, whatever ions you want to name. But we want to desperately reabsorb that glucose, and we do. We reabsorb virtually all of it. This is one of the ways that we reabsorb virtually all of it. The glucose in the filtrate, we want to reabsorb that. How we can do so is if we at the same time reabsorb that sodium. Make sense? Cool. If we halt the reabsorption of sodium, that means we halt the reabsorption of the glucose. And normally we wouldn't want that to happen. But in the case of type 2 diabetics, or just diabetics in general, but particularly type 2, right, it's not what first line, but one of the later medications that they potentially may put somebody on, something like Invokana, that halts, that decreases the permeability of the proximal convoluted tubule to sodium. So therefore, if we don't reabsorb sodium, that means we don't reabsorb the glucose, and more glucose is in the urine, and that means glucose levels in the blood decline. Make sense? Okay. So other things that we see over here. Right? Here's what's an example of an antiporter. Right? Again, these are two molecules moving in opposite directions. This says, hey, I'm going to trade this for that. Right? You go that way, I'm going to go this way. All right, cool. Kind of like nowadays with the social distancing. You go into the grocery store and you're like going head on with somebody down the aisle, and you kind of look at them with a stank guy, like, all right, what move are you making? It's like, all right, you kind of look at each other like, all right, you go that way and I'll go this way, and everyone will be okay. It's kind of like the Wild West. It's one of those great cheesy spaghetti westerns. If you haven't watched them, you got time now. Watch them. They're amazing. So these antiporters, right? When we talk about urine, right? When we talk about blood, when we talk about fluid inside of the body, right? The pH of that fluid is hugely important to how fast the metabolic reactions occur, how fast the chemical reactions occur. It is hugely important. One of the main ways that we can regulate pH, right, is through, again, over here, looking at the antiporters. Excuse me. Right here. So, how acidic something is, right? The greater the number of the number of hydrogen ions, excuse me, the greater the number of hydrogen ions, the greater the acidity. Meaning, over that scale of 1 to 14, 7 being neutral. So acidic is that way, right? So, for instance, when we look at the level of hydrogen ions in fluid that has a pH of 2, it is significantly higher than a fluid which has a pH of, or an acid that has a pH of 6. Right? So that's how we can regulate blood pH. Right? Okay, so here, right, for instance, if if hydrogen is entering into the lumen, right? If hydrogen is entering into the lumen, all right, the lumen, therefore that means that's going to be drawn from the blood, so it's going to make the blood a little bit more alkaline, right? Because remember that the range of blood pH, right? It's right around 7.35 to 7.45, so it's a very kind of small range. It's a very specific range. You know, in order to achieve this, right, it requires ATP, right? Absolutely, right? Again, these are all active transport. They all require ATP to actually function. Right? You can see, right? These sodium ATP pumps, right around 6% of the ATP that we utilize at any given time is devoted towards maintaining this pump, so we can therefore maintain our blood pH. So that's a pretty significant amount. I mean, 6%, right? That's a significant amount. anyways, you can see if, for instance, the blood is too highly acidic, we can again kick out a hydrogen. Right? If we kind of come down here, we look at right HTO3, which is a bicarb. Right? Either way, right? Kind of the big purpose to looking at these here is when we look at right the movement of sodium, right, as it relates to hydrogen. Right? Here we can see, right, sodium enters into the tubule, or I'm sorry, enters into the cell. Now, the movement of hydrogen here is going to continue to kick outwards. All right, so again, just kind of swapping this for that. Now, when we start to combine, there's something called carbonic acid that you want to talk briefly about in the respiratory system from last semester. Right? You would have talked briefly. You would have talked about that. That's where a large majority of the CO2, which is produced inside of the body, that's where it is converted into to help buffer the blood. Right? I mean, remember, as much as 70-ish%, give or take, right, is converted to carbonic acid. And the reason why that is carbonic acid is a very, very weak acid, but it helps to regulate the pH of the blood. Right, so as I had mentioned previously, all the CO2 that we produce, right, and every single time that we produce ATP, every single time we are utilizing molecules of oxygen, we are absolutely producing molecules of CO2. Right, so we're producing massive quantities of CO2. That's why it's so important. Right? It's not like CO2 is a bad thing, when yet 70% of it that we produce is purely functions to help regulate our blood pH levels. Right? So not all molecules are absorbed via active transport here, right? Absorption of glucose, right? Absorption of hydrogen, absorption of carbon dioxide, carbonic acid, right? That's all via active transport here. This is where we look at passive reabsorption. Right? Passive reabsorption, we see passive reabsorption in the proximal convoluted tubule. Now we can see, right, paracentral, transcentral, that just simply means, all right, does it go in between the cell, or does it go like right through the middle of the cell? And for the most part, ions can go either one. They can go between the cells, right, or kind of around, para, or trans, which means through. Right? Then we have osmosis of water, movement of water, same thing. But one thing to kind of pay attention to, there are channels which are specifically designed for water called aquaporins. Right? These aquaporins, they are water-specific channels, so water can quickly travel through the membrane. Yep, they are specific channels where, oh, you're water, awesome, you can travel through. If you're not, nope, sorry, no admittance is permitted. Right, kind of like those carpool lanes, which no one ever abides by, but on the highway around certain cities, right, they have those carpool lanes where if you have multiple people in the car, you can drive in that lane. If you don't, you're not supposed to drive in that lane. Same thing here. Right, these aquaporins are for water, and that's it. I don't care if you're chloride, it doesn't matter if it's potassium, calcium, glucose, doesn't matter. They are designed for water, and that's it. Right, that's all done passively. Right, that's all controlled just via diffusion. Right. So here, for the second part of the loop of Henle, right, two sections to the loop of Henle, or the nephron loop, right, or the loop of the nephron, all the same thing. Now the descending limb of the loop of Henle is very similar to that of the proximal convoluted tubule. It's essentially permeable to all ions in water, right? There's very little that it's not. There's very little that it doesn't permit. Right? The ascending limb. The ascending limb is, again, this part, and it starts to come up this way. Right? That's the ascending limb. Let's try that one again. Right, the ascending limb. Right here. Permeable to ions, calcium, potassium, etc. However, it is impermeable to water. Meaning water, if I'm water and I'm here, well, it's going to encroach upon the collecting duct. If I'm water and I'm here, if it's impermeable, that means I cannot enter into the tubule, and vice versa. If I'm water and I'm inside of the tubule, that means I cannot leave to re-enter the bloodstream. I am impermeable to water. Now, that's going to be hugely important a little bit later on when we look at how we adjust the concentration of the urine. Right? That's the important part. We'll look at that in a little bit. We can see, right, the ascending limb, right here, right? Whether it's chloride, sodium, potassium, calcium, etc. Right? They can all cross the tubule wall and enter into the ascending limb of the loop of Henle. Water is not permitted, right? We don't see water in this particular example. I guess think about it. If I'm water, I'm hanging out here. If sodium, potassium, chloride, right, calcium, whatever, you name it, leaves the tubule, enters into the vasa recta, what would water do? Well, water would follow. Water is a follower. Right? But water cannot cross the membrane. It cannot cross the ascending limb of the loop of Henle. So the water remains in the filtrate. Why that is, we'll talk about that in a minute. The distal convoluted tubule, that's the third portion of the renal tubule. Right? The function is very similar to that of the proximal convoluted tubule. Right? It has both symporters and antiporters, continues the reabsorption of ions. Its permeability, however, varies quite a bit. Right? Its permeability varies quite a bit. And this has to do with the fact of the amount of hormonal influence. Right? Whether it's antidiuretic hormone, whether it's aldosterone, whether it's adrenal peptide, right? There's a number of hormones which can go and impact, right, the permeability of water. Now we see regulator of blood calcium levels. Remember, in the talking about parathyroid hormone, talking about the parafollicular cells in the thyroid producing calcitonin, right? Those were hormones which helped regulate blood calcium levels. There was a hormone released from the kidney called calcitriol. Right? That helped to increase the absorption of blood calcium from the urine. Right? Or I guess more specifically, that helped to reabsorb the calcium in the filtrate, so therefore it wasn't in the urine. Right? This is where that occurs. This is where that hormone has its effect, right, within the distal convoluted tubule. So, final step, right, the collecting duct. This is the last collection port, if you will. Right? Any filtrate, anything still remaining in the collecting duct at the end, that is where urine is then basically dripped out and produced. Okay. So we see, due to hormonal influences, its absorptive and secretory abilities vary. Yep. Hormones like antidiuretic hormone. Right? antidiuretic hormone that can greatly influence how much or what the permeability is to water, for instance. We're going to talk about that here in about 1 second. All right. So when we look at this, okay, these are all hormones we've mentioned before: parathyroid hormone, angiotensin 2, atrial natriuretic peptide, aldosterone, antidiuretic hormone, again also called vasopressin. Let's look at aldosterone right first, because that one we talked probably the most about. We just talked about it last class. We see it's secreted in response to decreased blood volume, which causes a reabsorption of sodium, as well as increased secretion of potassium and hydrogen. Okay. So one of the things with this, right? So if let's just forget this for a second. The reason why I didn't mention it, right? I previously hadn't talked about it. But when we look right here, sodium reabsorption, hydrogen secretion. Remember those antiporters? These guys here, right? How sodium goes one way, hydrogen has to go the other way. That's why we see with aldosterone, the way that aldosterone functions, aldosterone, I'm just going to draw some kind of red squiggly lines around here. Let's imagine these red squiggly lines represent the blood vessels, right, the vasa recta. So the reabsorption of, with aldosterone, the idea is to retain fluid. We want to reduce the amount of urine we produce. So how do we do that? Well, water's a follower, so if I can pull more sodium from the tubule into the bloodstream, water will follow. Does that make sense? Right. But when we see, right, because of the fact that sodium and hydrogen, they act as antiporters, meaning if sodium goes one way, hydrogen has to go the other way. So it does. As sodium enters here, hydrogen is kicked into the tubule. Right, and I just realized where I was drawing this arrow here, so let me move that in case it's confusing people. Right. If sodium leaves the tubule, again here it's drawn the other way, doesn't matter. Here, sodium leaves the tubule because of the fact that it's an antiporter, right, or it's the antiporter process. We have to kick out a hydrogen, meaning we have to secrete a hydrogen, so hydrogen levels are going to drop. Right. That's why we mentioned, that's why along the proximal convoluted tubule, along the distal convoluted tubule, along the collecting duct, the permeability and the molecules which are gotten rid of drastically vary because of the amount of hormonal influence. Okay. Here, antidiuretic hormone, secreted in response to increased plasma osmolarity and decreased blood volume, binds to receptors and principal cells, causing increased number of aquaporins and H2O reabsorption. Wow. Just previous slide. Right. By previous, I meant like 6 slides ago. These aquaporins, they are channels specifically for water. Right. That is how ADH, or antidiuretic hormone, functions. With antidiuretic hormone, okay, we want to reabsorb as much water as possible, so water goes from the tubule and leaves and re-enters into the vasa recta, re-enters into all of the capillary beds. Parathyroid hormone. We talked about this previously. Right? Endocrine system in a little bit now. Secreted in response to decreased calcium levels. Yeah, remember, parathyroid hormone functions to increase calcium levels. Right? If our blood calcium levels are low, or if they're lowering, we're going to have secretion of parathyroid hormone. Right? So we can see where that impact or where that takes place. And you can see impacts along the proximal convoluted tubule as well as along the distal convoluted tubule. Right? It increases the permeability of calcium, therefore more calcium is reabsorbed. Again, that's the parathyroid hormone or parathormone. Angiotensin II. Right, last class, when we talked about the renin-aldosterone pathway, right, angiotensin II, that was one of the kind of precursors to eventually producing aldosterone. All right, remember, the liver produced angiotensinogen, the kidney produced renin, they had a baby, which became angiotensin I. Angiotensin I, thanks to angiotensin-converting enzyme in the lungs, became angiotensin II. And then angiotensin II then went and caused changes in vasoconstriction, vasodilation, as well as ultimately led to the adrenal gland to release aldosterone. So we can see angiotensin II, released in response to decreased blood pressure, causes efferent arterial constriction. Well, yeah, think about it. Remember GFR, right? That's right. Really, when we impact GFR, that's what impacts everything else, right? Like if we impact the rate in which the glomerulus is functioning, that can impact how much absorption and secretion is occurring. So if our blood pressure is low, how much fluid or how much filtrate is being produced? Not much. Okay. Well, that's a problem, right? We want to ensure that we produce as much filtrate as necessary. Right? We want to make sure that the filtration rate doesn't drop too low or that it doesn't drop too high. Right? But as blood pressure drops, so does GFR. It drops too low. We're not producing enough filtrate. If we're not producing enough filtrate, that means we're not going to reabsorb sufficient levels of, ions, of amino acids, etc. So what happens? Angiotensin II, you can see right here, actually leads to a constriction of the efferent arteriole. Like, whoa, huh? Doesn't impact the afferent arteriole, but it constricts the efferent arteriole. Think about it for a second. What would that do to GFR? Alright, well, we said previously with the sympathetic motor neurons, they can, if the level, if GFR is elevated, or if there is too great a volume getting into the glomerulus, we can constrict the afferent arteriole to allow less blood flowing into the glomerulus. This is kind of like that, but it's the opposite. But as far as involving pressure and volume, it's the same concept, right? Even though we're not constricting the afferent arteriole, which we wouldn't want to do, right? It's not like we can make it dilate beyond its capacity. But if we constrict the outboard, the efferent arteriole, remember, that's what carries blood out of the glomerulus. So remember, the afferent carries blood into the glomerulus, right? Blood's flowing around, producing filtrate, producing filtrate. The leftover blood then leaves the glomerulus via the efferent arteriole. So when it leaves the efferent arteriole, if we constrict the efferent arteriole, just think about it. What does that do to the blood within the glomerulus? Oh, it's going to think of it as like, oh, it's going to kind of going to kind of backflow a little bit. What does that mean? Oh, it's going to increase the pressure inside of the glomerulus. Well, that's a good thing because the blood pressure was too low to begin with. Right, so if our blood pressure is too low and not enough water is getting into water, if not enough blood is getting into the glomerulus, that means the pressure is insufficient. Right, think about filling up a 5-gallon pail that has a crack in it. Right? Well, whatever, you've got a spigot that's broken and you can only turn it on just a little tiny bit. So you turn on that spigot and you're starting to fill up the bucket, but the problem is there's a big hole in the bucket. Alright. So you're filling up the pail, water's leaking out. That's kind of the same idea here, right? Blood's filling up the glomerulus, but then the blood is leaking out. It's supposed to. Well, I can't let the water, my problem is the flow of water, because of the pressure is so little, it's not filling up the bucket, right? It's not filling up the glomerulus. So I can't increase that. What can I do? Well, I can close off the crack in the bucket, right? I can pinch off, or I can seal up that crack, so therefore the bucket starts to fill up more and more and more. Same idea here. Obviously, the efferent arteriole doesn't constrict entirely, but it significantly constricts. Therefore, less blood exits the glomerulus, more blood remains in the glomerulus, therefore that then increases the pressure inside of it. Yeah? Make sense? Okay. Last but not least, right, it's kind of unique, right? All of these hormones, it kind of shows you the importance. But for angiotensin 2, parathyroid hormone, ADH, aldosterone, right? They are all functioning to elevate volume, elevate pressure. Atrial natriuretic peptide is just the opposite. Right? We see with secreted in response to increased atrial pressure, right? This causes increase in GFR. So if we have too much volume, too much blood volume, all right, if we have too much blood volume, that means that we need to produce a greater amount of urine so we can get rid of it. Because if we have too much blood volume, blood vessels are distending too much, the chambers of the heart are distending too much. Right, so in the right atrium, in the wall of the right atrium, there's cardiac cells which actually release ANP, something called atrial natriuretic peptide. Right, you can see where that works. That functions along the distal convoluted tubule. Right. We can see, right, causes increased GFR and increased sodium filtration with no compensatory sodium reabsorption in the distal nephron. Whoa, what the hell does that mean? What that means is sodium is not allowed to be reabsorbed. Right? If sodium is not allowed to be reabsorbed, that means sodium remains in the tubule. If sodium remains in the tubule, where does water remain? Water remains in the tubule. If water remains in the tubule, it remains in the collecting duct, and now it is destined to leave. Right? Now it's destined to become lost in the urine. Alright, so, and that's something to kind of recall or to remember, right? When the water, right, is higher in concentration in the tubule, right, water is a follower. If we need to have elevated, right, blood volume, or if we needed to have, yes, all right, sorry about that. My my youngest, she needed my assistance to say the least. So I apologize about that. Anyways, from here, right, the renal tubule, right, the effects of the hormones on the tubule. The entire reason why, right, and it's not like, oh, there's just some random secretion of antidiuretic hormone or aldosterone just like once or twice throughout the day. This is an ongoing kind of never-ending process, depending on what you've just consumed, right, your activity level, right. These, if you just ate whatever, a kale, again, hypothetically here, you just ate a kale salad, so you just intook a bunch of calcium and a bunch of magnesium and a bunch of, potassium and those sorts of things. You're going to get rid of greater amounts of those in the urine, right? So again, that's where, what you eat greatly dictates the physiology and the ions, molecules, nutrients that are present and what has to be gotten rid of, right? So we can see, right, what is, right, the actual kind of influencer. What is the actual greatest dictator of how much urine are we going to produce? Right, we can see hormonal regulation and salt loss, drinking salt water. Right, yeah, just think about it. Right, one of the most important influencers of our body's overall hydration levels is the amount of salt that's lost. When we say salt, I mean, yeah, ions, sodiums, the solutes, because water is a follower. Right? Water is going to follow those solutes. That's the issue with drinking salt water, for instance. Hey, I'm on at sea. I'm on a deserted island. A boat crashed. Whatever. I am on Tom Hanks and Castaway, which, geez, that came out in 01, I think. So 2000, that came out before some of you were even born. Good God. Anyways, right, drinking salt water, terrible, horrible idea. You will dehydrate way faster than you would if you didn't drink any water, right? Because if we ingest a large amount of salt, we need to get rid of a large amount of salt. Well, the only way we can get rid of large amounts of salt is if we also get rid of a lot of water with it. It's not that we want to get rid of water, we want to retain the water. And before you're like, well, isn't there a lot of water in salt water? Yeah, there is. Right? But even if we drink a gallon of salt water, we get rid of greater amounts in order to get rid of the amount of salt that's in that water. That's how much salt is in that water. I'm not knocking salt. Salt is fantastic. I mean, when we've talked about function, salt is, it is as important as glucose is, right? So, right, salt is salt is okay. However, in salt water, right, like I mentioned, significant quantities. So that's the biggest influence, is how much salt is actually lost through the urine. Right? So if you want your body to retain fluid, the urine will be concentrated. If you want to get rid of fluid, the urine is going to be dilute. So up to this point, we've been talking about, something called a countercurrent multiplier. I never actually used the term of that before. But we see the flow of solutes dictates the direction of water. Right? Both together greatly regulate the flow of water as well as the concentration of the urine. Yep, so along this entire process, along the tubule, why it's important for along the proximal convoluted tubule, along the loop of Henle, along the proximal convoluted or the distal convoluted tubule, and the collecting duct. The reason why certain molecules can and can't cross has entirely to do with our kidney's ability to concentrate the urine. Right? If we need to retain a large amount of fluid, right, the urine is going to be very concentrated. You're going to look down and say, oh, it's like yellow, orange. Well, no, orange, that's severe, but right, it's going to be a very dark color. It's very concentrated. If it's very, just drank a bunch of water and had to pee a little while later, and I look like, oh man, it just looks like water. That's an example of a very diluted urine. Right? And that has to do with, again, the movement of the molecules. remember like concentration gradient, diffusion, all that kind of stuff. I'm going to briefly cover diffusion for about 3 minutes. Well, I say briefly for 3 minutes. Briefly, 3 minutes I don't think is brief for diffusion, but anyways, when a molecule, right, goes from an area of high concentration to low concentration, so high to low, right, that's an example of diffusion, and that keeps happening until eventually the levels equal out. So let's just, for example, let's say sodium levels are super high here, right, in the fluid in the peritubular capillaries. Well, it would re-enter into the tubule. Right. Let's just imagine, calcium levels are super high in the ascending limb. So they leave and enter into the capillaries. Right, so you always got to think about the movement of these ions. It is going to change, right? For instance, let's imagine here the red just represents sodium. All right, so as sodium, let's just imagine sodium leaves. It's reabsorbed. What did water do as well? Oh, water also left, right? Water left. That's why you see the concentration increase, right? From 300, 350, 550, 750, 900. No, you do not have to imagine or remember these numbers, not at all. All right. But as water leaves, that increases the concentration of the urine. Right? All right. Now, as we start to go up the ascending limb, think of it. Well, if the concentration, because this all happens very quickly, as the concentration increases, what did we just talk about with diffusion? That's when molecules go from an area of high concentration to low concentration. Oh, all right. So if it goes from an area of high concentration to low concentration, as it begins to elevate, you can see how solutes molecules begin to leave. Right? As they leave, the urine becomes less and less concentrated. Right? Just think about it. Let me erase these arrows for a second. At this point, the urine's super concentrated. High concentration of solutes, sodium, potassium, waste products, etc. So the ones that are permeable are going to leave. And that's when you see, again, 900, goes to 550, which goes to 350, which goes to 150, which goes to 100. Right? We will never see this number elevate. It just keeps on dropping. And why is that? Remember, part of the collecting duct is it is at this, without the presence of antidiuretic hormone, it's impermeable to water. Right? And really, it wouldn't matter if it was, right? Because of the fact that the concentration, right? The concentration begins to decrease of the solutes. Right? So does that make sense? Let me explain the kind of the concentrated, right? How we kind of concentrate the urine, right? It might make a little bit more kind of sense to you. Yes, Claire. All right. So my sincere apologies. I had to pause and step away for a moment. My youngest is she loves her daddy. What can I say? So, anyways, I'm gonna kind of back up and kind of just cover this again from kind of the start. Depending on our hydration levels, right? How much we drank, how much food we ate, right? Because really, by and large, the amount of hydration that we have, or our hydration level, is much more tied to food and the type of food that we eat than the actual amount of water that we drink, right? The food that we eat, most food that we eat has a pretty substantial amount of water component to it, right? So depending on how much we've ingested through the food, through liquids, how much we've lost through sweating, through activity, let's ignore hemorrhaging, like, oh, I just lopped my leg off. Let's just ignore those, right? In times where we don't have appropriate hydration, or we want to retain as much fluid volume as possible, right? In order to make sure that filtrate movement is always through the tubule, right, like this. There's not like cells, right? There's not like muscles surrounding this. It's not like in peristalsis, right? Where in the digestive system you swallowed, the smooth muscle kind of pinched and constricted and then pulled and propelled that food down in the stomach. It doesn't undergo peristalsis like that. So the movement of this filtrate, the movement of this fluid, is based upon the molecules. All right, get rid of this line. So in concentrated urine, we can see come down. Again, we can see, right, as the water, as we begin to descend down, once we get into the loop of Henle, right, we can see how the concentration is increasing. Right. Part of that is because the amount of ions has fluctuated, right? Whereas in the proximal convoluted tubule, right, that's essentially permeable to everything. Right? The loop of Henle is where it starts to kind of dictate a little bit as far as what can and cannot get through. Right? For instance, the ascending limb, impermeable to water. Right? The descending limb, permeable to most salts, most ions, most water. There are some solutes which aren't. Hence the reason why you see, let me get this line out of the way. Hence the reason why you see it goes from 300, 380, 580, 780, 980, 1200. It's very, very concentrated. Okay. As we're starting to go up here, ignore that thing, I'm not sure what, I don't know, the computer just had a stroke or something. Water cannot leave, right? Nor can it re-enter, right? This is a wall. This is impermeable. This cannot, right? No water can penetrate this. Okay. As it ascends, we can see how the solute concentrations are actually beginning to decrease, which doesn't make a whole lot of sense because of the fact that, right, once concentration levels start to decrease of the solutes, water can't move, so this shouldn't actually continue to happen. But right here, presence of these symporters. Remember, symporters, there's an active transport, meaning that's where we're actually pumping molecules using ATP. Meaning, so excess ions of sodium and chloride are being pumped out into the fluid here. Now, that in and of itself right here isn't that important, right? It's not like that causes the movement of, through the ascending limb. Where that's important is, think about it, right? If chloride, sodium, right, are all being dumped into this interstitial fluid, well, what happens to the water in the collecting duct? We can see this is what actually pulls the water from the collecting duct. Right? It pulls more and more and more fluid from the collecting duct. So what happens? We're now left with a concentrated urine. This one specific part of the ascending limb specifically pumps out the sodium and chloride ions, more than they should. Right? So there's already a heightened concentration of sodium and chloride within the interstitial fluid. That's what draws the water into the fluid. Does that make sense? And like, well, the solute concentration just keeps going up. Yeah, solutes aren't a follower. Right? Water's a follower. So water leaves, which, yes, starts to increase the concentration of the solute, absolutely. Right? But not more so than what the concentration of the interstitial fluid is. Right? This interstitial fluid, it's super concentrated already. So that's what pulls the water into the interstitial. Right? So once that urine is to be stored, we've produced the urine, we now need for it to go from the kidney, collects in the ureter, then from the ureter it's transported into the bladder. Right, from the bladder, that's where it's then stored. Right, all the different specific structures here in lab, you cover those, so I'm not going to cover them here. Right, but the ureter, or the urinary bladder, that's where it's stored. Right. You can see each ureter, right, undergoes basically peristalsis to help kind of push that urine down into the urinary bladder. Now, granted, right, pretty much gravity alone would take care of this, right? You can see capacity around 700 to 800 milliliters. So, onto the actual reflex, the actual kind of process, what's called the micturition reflex. It's all done via a reflex. Right? As the bladder fills, it distends. As it distends, little touch receptors, stretch receptors, right, enter into the spinal cord. Now, as they enter into the spinal cord, right, sorry, I'm going backwards. This one here, green one, right, this is the sensory neuron carrying that sensation from the stretch receptor into the spinal cord. Awesome. Remember the specific part of the spinal cord, posterior gray horn, right, that initiates this interneuron, which then goes to the ventral gray horn. Now, remember, even though this is a spinal reflex, and this reflex initiated in the spinal cord, the brain absolutely knows what's going on. Right? The brain has absolutely initiated and informed what's going on. That's why you're able to stop yourself from urinating, for instance. Right? If your brain was just left out of the loop. That's why, like, brain injured people who have certain neurodegenerative diseases and conditions, they have issues regulating bowel and bladder function, right? Because let's just imagine if this didn't occur. Well, that reflex is still initiated. When that reflex is initiated, that then travels down to the smooth muscle of the bladder and causes it to contract. As it contracts, the opposite happens, right? As it contracts, urine is then expelled into the urethra and outwards. We can stop that, fortunately. Right? This doesn't have to happen. It's not like, oh man, I'm getting the urge, I'm going to pee. All of a sudden, you don't just then pee. All right, what happens? We can actually stop the flow. And that is where the involvement of the brain comes into play. Right? This right here, this is the external urethral sphincter. This is under voluntary control. This is skeletal muscle. You can contract this. This is, oh man, a great part of Dumb and Dumber. I came out in '94, so that was before most of you were born. But it is an unbelievably great watch. It's a ridiculously funny movie. But there's one part where Harry and Lloyd are on this trip, and Lloyd's got to pee, can't stop himself. He ends up filling up like 8 bottles of 8 empty bottles like with urine. And he's like, I just can't stop myself, man. we do have the ability to stop ourselves, right? We do have the ability to halt, right, urination, right? Just like that's why you first get the idea, I'm gonna pee. All right, you don't just immediately just go. You stop yourself and then make it to the tree, the toilet, Buddy's gas tank, whatever it happens to be, right? But that's that first initial kind of, I gotta pee. What is that? Well, the sensory neurons went into the spinal cord. Right? The spinal cord initiated the interneuron, send it up to the brain. Right? Once it hits the brain, that, I gotta pee, that's where that initial sensation comes from. Right? Message from the brain comes back down, goes to the ventral root, out to the urethral sphincter, right, or the external urethral sphincter to contract. Therefore, we don't initiate urination prior to when we're actually supposed to. That makes sense? Okay. All right. Again, the anatomy here, the different portions of the urethra, again, covered all that in lab. I'm not going to cover it here. But one of the things, right, we see urethra, right, in males is around 5 times the length, right? It is substantially longer in males, right? It doesn't matter endowment, right? The urethra is just substantially longer, right? I mean, the female urethra, a few inches, right? Men, it's upwards of a foot, right, through the entire phallus, through the base, all the way up through the prostate. I mean, it's just a much longer course that it has to travel. So because of that, that's why urinary tract infections, kidney infections, those sorts of things are definitely much less common in men, right? Especially bladder infections, right? Because it's just a distance game. The odds of me walking 100 feet today, pretty good. The odds of me walking 100 miles today, not a chance. Right? Same thing with bacteria. Bacteria getting its way, swimming, crawling, actually getting its way into the bladder of a male is pretty slim. Looking at the distance it has to travel, look at comparatively to that of a female, the urethra is substantially shorter. That's why young girls, especially learning how to wipe properly, proper hygiene, urinary tract infections are quite common. Right? I mean, just think of proximity, right? As far as rectum, urethra, I mean, it just had just a kind of a proximity game, if you will, right? So. Anyways. Last thing we'll kind of cover here, again, looking at kind of evaluation of the kidneys, looking at some different conditions, some different problems which might arise from the kidneys. And one of the ways that we assess, right, the kidneys is through a urinalysis, right? Urinalysis, and just think of that as being a urine analysis, right? Now, when we think about what we can do, what we can assess, we see glucose, proteins, white blood cells. Those are just some of the things, right? But glucose, someone who there isn't supposed to be glucose in the urine. There's just not. Diabetic, especially uncontrolled diabetic, they're going to have glucose in the urine. Now, why? Because urine or glucose is in the tubule, absolutely. But thanks to these symporters and whatnot, our body can absorb glucose, but it's not infinite, right? Our body can absorb 160, 70-ish grams of glucose, right? So if we have more than that, uncontrolled diabetic, it's much higher than that, right? Then we can't absorb that much, so it has to remain in the tubule. So if it remains in the tubule, that's where it then remains in the urine. Do a urinalysis, say, hey, you have glucose in the urine, do a diabetic workup, right? Or some other pathologies going on. Something else, right? Proteins. As I mentioned, there is in post, some major surgery, in puberty, right? A small amount of protein in the urine, first thing in the morning is relatively normal, right? But proteins, large amounts of proteins in the urine, that's problematic, right? That's an issue. That's abnormal, right? That has to do with, well, if the pores, if the fenestrations in the renal tubule, right, are enlarged, that permits larger degrees of molecules or larger sized molecules through those pores. If larger molecules can get through the pores, then those molecules will end up in the filtrate. The other thing that we talked about was the fact that protein levels are what help regulate where the fluid is going to be. Is it going to be in the interstitium? Is it going to be in the bloodstream? Right, and that's where we talked about people who have edema, or people who are going into liver, liver failure might as well, liver failure, renal failure, right? They might end up with edema, and the problem or the reason as to why that is these proteins are produced in the liver. If our liver can't adequately produce these proteins, now we can inadequately regulate where that fluid is in the bloodstream or in the interstitium. Same idea here with renal disease. If greater amounts of proteins are being lost in the urine, that means there's going to be less protein in the blood, right? Because the kidneys can only I mentioned this 3 or 4 times. The kidneys can only filter the blood. Kidneys can't filter the interstitium. They can't filter that fluid between the tissues. So if you want the kidneys to filter the blood, right, or if you want the kidneys to filter the proteins, they have to be in the blood. So if the kidneys, even though they're not supposed to, if there's renal damage, some sort of infection, proteins being lost through the urine, that means there's less protein in the blood, and that means there's less kind of drawing force of that fluid, that oncotic pressure from the interstitium back into the blood. And we talked about this on the previous presentation. And lastly, white blood cells, right? Let's say you have some sort of urinary tract infection, bladder infection, whatever, what have you. That's where you might end up having white blood cells within the urine itself, where you have those immune cells. Now, cells in themselves, they're too large for to fit through the fenestrations, to fit through the pores. But if they're already in the tubule because of some sort of tubular infection or something like that, then it's irregardless because they doesn't have to fit through the pores or the fenestrations. They came directly through the tubule. Now, with renal hypertension, right, we've all heard of hypertension, high blood pressure. Oh man, everybody's got it. Everybody knows their son. Right? Renal hypertension is essentially when the hypertension itself is being driven from the glomerulus, or from the kidney itself. All right, so let me back up to this, and let's go to this one. Come on. There we go. So remember, as blood comes into the afferent arteriole, cool. What happens if the afferent arteriole constricts? If it constricts, oh, okay, we allow. Actually, let me just draw 2 to begin with. If the afferent arteriole constricts, now we've got less blood flowing into the glomerulus. Okay, cool. If somebody has just hypertension in general, that's one of the doesn't have to be, but that's one of the things which can lead to renal hypertension. Think of it. If somebody already has higher than normal blood pressure, great. That means that greater amounts of blood will be flowing into the glomerulus. We need to stop that, right? We can't have elevated GFR. We can't have elevated glomerular filtration rates. So remember angiotensin 2? Right? That can help to constrict the efferent arteriole. Now, when this starts to happen, you can envision, if this constricts, that starts to backflow blood up into the glomerulus, and that's going to increase the pressure inside of the glomerulus. One of the problems when we think about this, all right, so if that's the case, if we're increasing the pressure inside of the glomerulus, I can only, right, the afferent arteriole, all I can do is constrict it, right? I can't cause it to dilate, for instance. It can only get so big. So because of that fact, right, one of the ways to increase the pressure inside the glomerulus that we had talked about is because of the constriction of the efferent arteriole. So if we have, because of the high blood pressure, that's going to cause the afferent arteriole to constrict, which will reduce the GFR. But the response to that is to constrict the afferent arteriole, which then, as it constricts, that increases the pressure inside the glomerulus. But people who have sustained hypertension, that's going to continuously cause the afferent arteriole to be constricted, which would in turn then cause the efferent arteriole to be constricted. So you can just envision how the entrance and the exiting, or basically if we have less fluid, less blood being able to exit than enter, that's where we have renal hypertension. Right? Like just think about it for a second. You have heightened blood pressure inside of the glomerulus. Right? If you have increased pressure inside, if you need to increase the pressure within the glomerulus, again, that's where we can reduce the efferent. But because of that, that increases the pressure. Right? So again, just think about that for a minute. I am already over time. I've probably got about 15 minutes left as far as the renal diseases. So what I will do, I will end this presentation here, and I will pick up with this next class. I'll probably start entirely new from this slide as well, but I'll certainly be focusing on glomerulonephritis, kidney stones, and UTIs, and I'll readjust renal hypertension as well. But I'm already a few minutes, so everybody take care. Have a great weekend, and stay safe.